Hello, my name is José Manuel Sánchez Guardado. I am a master student at the Universidad de Guanajuato, and the topic that I come to present today is near infrared emission of erbium-doped non-cytotoxic calcium aluminate inside tier biological window. Since the invention of X-ray and its use in medical diagnostic, the creation of modern non-invasive techniques can be defined. However, some of these, as well as magnetic resonance, expose the human body to high doses of radiations that limit the use in treatments. With its why new efforts have focused on fluorescence imaging, using nanoparticles, usually made of high-pressure metal and semiconductor ceramics, but which have expensive synthesis methods, limiting their use. For this reason, this work seeks to obtain lower-cost alternative based on the use of calcium aluminate by simple rod doped with erbium to take advantage of its emission for the tier biological window. The synthesis of pure and doped calcium aluminates with different concentration of erbium was performed using the simple combustion method. Aluminum nitrate and calcium nitrate were used as precursor for the host. In an aluminum-calcium ratio of 2 to 48 that allows the graphite phase of the calcium aluminate to be obtained, while as precursor to the dopant, erbium nitrate was used in a ratio of 0.01, 0.05, 0.1 and 0.2% molar. An aqueous solution was prepared, adding nitrates in the ionized water while maintaining under a constant agitation until complete dissolution. Then urea, ammonium nitrate, and boric acid were added as flux to achieve a complete full oxygen ratio. The solution was kept in a constant agitation for 30 minutes until a clear solution was obtained. Subsequently, rapid self-sustaining combustion of the solution was carried out in a preheat muffle at 600 Celsius degree for 5 minutes. A low-density foam of a pale pink color was obtained, the intensity of which is increased with the concentration of air. The foam was ground, obtaining a fine powder. The American Tissue Culture Collection Human cervix adenocarcinoma cell line was used for the cytotoxicity study. 20,000 cells per well were grown in a Dubalcus minimal essential medium to 1% antibiotic antifungal for 24 hours in a 37 Celsius degree in an air carbon dioxide atmosphere. The solution were prepared for the evaluation of pure and doped samples with the following concentration: 5, 2.5, 1.25, 1, 0.6, 0.3, and 0 milligrams by milliliter of each samples. Cell viability was determined by the violet crystal assay. The absorbance was measured at 595 nanometers. The data was standardized with respect to the control. The diffractograms of the pure calcium aluminate materials antidoped with erbium are shown in the figure. All diffraction patterns have the same set of peaks, belonging to the graphite phase. All samples have irregularly shaped polycrystalline particles with a high porosity resulting from the rapid and violent release of gases resulting from the combustion synthesis process. The distribution of particle size for all samples is extended from 1 to 100 micrometers. The 0.2 molar calcium aluminate absorption spectrum of erbium is shown in the figure. An absorbance band below 350 nanometers corresponding to the crystal lattice is observed, while a series of bands above 360 nanometers to 1650 nanometers are seen corresponding to the energy levels reported for the erbium ion. The data show that the absorption band centered at 800 nanometers has greater absorption cross-section than the typically reported for the erbium. This increase makes excitation of the material within the region of the first biological window. This figure shows the near-infrared emission spectra of calcium aluminate samples, pure and doped with erbium. As expected, the pure host has no emission when excited with a wavelength of 808 nanometers. The emission observed in the near-infrared region in all doped samples corresponding to the transition 4E13½ to 4E15½ of the erbium centered at 1.5 micrometers. It is observed that the intensity of the 1.5 micrometers emission increase as the erbium doped increase. This increment is not linear. The relationship between the intensity of the emission with the erbium concentration shows a trend that closes the saturation for 0.2 molar percent of erbium. The figure shows the toxicity analysis of pure and doped calcium aluminate samples with different concentration of erbium. Cytotoxicity induced by different doses of pure calcium aluminate and doped with erbium was not observed compared to the control group. Most samples showed a tendency to increase in the percent of cells when some doses were used. However, they showed no statistical significance. On the other hand, the calcium aluminate doped at 0.2% molar of erbium was the sample that showed a marked tendency to decrement in the viability of HeLa tumor cells, since in most of the doses used a smaller number of dead cells were quantified. This decrement did not achieve statistical significance with respect to the control. In general terms, the morphology and crystallinity of the sample synthesized by different concentration of the dopant did not undergo any significant change. The main influence of the calcium aluminate matrix was observed an increase in the not very sensitive absorption band of the 4A9½ energy level for erbium, 5 times higher than the reported in the literature. This allows the excitation of the samples within the first biological window and emission in the third biological window. Another result to highlight is that the samples do not induce acidotoxicity to HeLa cells, which is suggested to the harmless material, which do not cause damage or change in human cells. Photoluminescent results coupled with the cytotoxicity analysis make the graphite phase of erbium-doped calcium aluminate a potential candidate for a wide variety of biomarker applications. Thanks for your attention.